Hi guys, welcome to genetic fingerprinting. So you need to be able to describe what genetic fingerprinting is, explain the technique of gel electrophoresis, explain how genetic fingerprinting is carried out, explain how the results of genetic fingerprinting are interpreted, and consider the uses of genetic fingerprinting. So this is our specification. So there is a lot of things that you should be able to do looking at the data provided from genetic fingerprinting. And the uses of the genetic fingerprinting were in the learning objectives are, of course, the forensic science, medical diagnosis, animal and plant breeding. So that's one of the learning objectives done. And let's have a look at the rest of them. So what is the gel electrophoresis used for? Is used to separate DNA fragments according to the size. So we need to remember that smaller DNA fragments travel the furthest distances. Okay, so it's not faster, it's just travel the furthest distance. So what's another important thing is the fact that DNA is negatively charged, and how the gel electrophoresis can separate the proteins. So apart from depending on the size, which we've mentioned, but also depends on the charge of protein. So those two things will apply in your past paper questions. So another definition before we will look into the process, it's the VNTR. So most of the eukaryotic genome contains many repetitive non-coding bases of DNA. So we call them variable number tandem repeats, VNTRs. Where are they located? They are located in a genome where a short nucleotide sequence is organized as a tandem repeat. So in terms of VNTRs, they are unique. So there's a unique number and length, unique pattern in every individual. So apart from the twins, everybody will have a different pattern of VNTRs. So if there are organisms, individuals closely related to each other, they will have more similar VNTRs. So let's have a look at the process of the genetic fingerprinting. So it's made of 5 stages: extraction, digestion, separation, hybridization, and development. So let's have a look, how does it work? So we're starting with the extraction of DNA from the sample. So if the sample is really small, we can increase the number of the sample using PCR. So in one of the previous videos, you can look up for the method of PCR if you are not aware of it yet. Okay, so extraction using extraction is the extraction of the DNA sample, and if it's too small, you can use PCR. But if the sample is too big, you can cut it using restriction endonuclease enzyme. Okay, so we can cut close but not within the targeted DNA. So those are two options. Okay, and then it's the separation. So we did mention that DNA is negatively charged and will move towards the positive charge. So there are fragments of DNA are separated using gel electrophoresis. So we did mention that they are separated according to size. So the smaller fragments travel the furthest distance, while the large fragments will travel shorter distances. And this is this could be also separated by the charge, and a double strand will be separated here into the single strand due to the alkali applied to the gel electrophoresis. So separation is taking place. So those our DNA fragments will be then transferred from the gel to nylon membrane. Why we're doing so? Of course, we need this reading to be accurate, clear, not on the gel. So next step is the process of hybridization. So what do we do here? We are going to now add our DNA probes. So in our previous video, we learned about the probes. So we can have radioactively labeled probes or fluorescent labeled probes. So we can use those to bind with VNTRs, because what is a DNA probe? It's a complementary single-stranded DNA which will bind to this VNTR. If they are not complementary, of course, it's not going to bind. So how then we can identify them? So the next process is called development. So the membrane with radioactively, for example, labeled probes will be placed on the X-ray film. So this is why we've used the nylon membrane, because we could not do it with the gel. Okay, so if so development of this X-ray film will show us the dark bands, okay, and this is where the radioactive probe has attached to this complementary fragment of DNA. So the film is exposed, and the points correspond to the position of the DNA fragments as separated in gel electrophoresis. And of course, we know that the pattern will be unique apart from the twins. They're going to have the same identical twins. They're going to have the same pattern. So to summarize here, it's the order activity for you. So the task is to describe how genetic fingerprinting is carried out, and those sentences are not in the right order. So you can pause this video for a second and you can order events to show the process of genetic fingerprinting. Right, ready for the checkpoint? Okay, so you should have DNA. It's extracted from sample. It's cut into fragments using restriction endonuclease. It will that will require the course sequence. DNA fragments will be separated using electrophoresis, and you could say about the process a bit more detail, including the electric current being passed through. The gel in alkaline solution was applied to separate DNA strands. Cover with nylon membrane, because we remember nylon membrane was used. DNA is fixed to the nylon membrane using, for example, UV light. And radioactively marker or probe would be applied, and this will be identified under the X-ray film if the bands, the present bands, right? So that's everything for genetic fingerprinting. See you later.